what. But fortunately, in this group, I think there is no problem, and most of you are present right now. Right now, 19 students are present in the lecture, so it is good. But because both groups are ongoing in the same program, the most probable today we have a shorter lecture, something around 50 minutes to 1 hour, and we will continue next week, both groups in the same progress. So, semester, so you had many lectures with me, and without any problem, you passed all of them. So this semester, you don't have any problem, and you have sufficient time, and many pages are repeating with different figures, with the same figures, repeating and repeating to understanding well. So, for example, if you have 10 pages in one lecture, most of them are repeating of the text and same photos. Don't worry. Today, we want to start. Okay, you shouldn't forget that we talk about examination 1 week before exam. Through discussion related to the how to have the examination, whole the semester we should talk, and this talking about the examination is endless. Will be finished. So at the end of the semester, 1 week before examination or 1 week before finish the lectures, we can talk much about examination. Yes, we'll do it. Okay. By the way, so today we want to talk about another bacterium with the name of Bordetellapertosis. Bordetellapertosis, which is the causative agent for a disease with the name of whooping cough. Whooping cough. The most probable you heard or you see some patients with this problem. And when a physician asks lab to do an examination, diagnostic test for identifying this pathogen, cough plate method is used. Cough plate method, which a plate to be placed in front of the mouth of one patient, and the patient have many droplets and aerosols come out from mouth going to the surface of our plate, and then after incubation, checking for growth and bacterial identification. But the main thing which it is the time to focus on is the pathogenicity and toxicity of this bacterium. As we understood, the main target of this bacterium is lung. And produce pulmonary infection. Normal case, when we see the lung normal ciliary movement is there. But when Bordetellapertosis is present in the lung, they go through, they can be located between and by bacteria, Filamentus hemagglutinin, as well as Pertussis toxin, responsible for stop motion. They're responsible for cilia and make the cilia in a static situation. So, according to this bacterium, Bordetellapertosis, which have great virulence factors, be the causative agent for severe infections, especially in lungs, with severe pulmonary. In this photo, we can see the synergy between Pertussis toxin. This is toxin, yes? This is Pertussis toxin. You can see this is everywhere. Pertussis toxin is present. And Filamentus hemagglutinin, this is Filamentus hemagglutinin is there. And binding to ciliated respiratory epithelial cells, yes, they can bind to ciliary epithelial cells by toxins, by Filamentus hemagglutinin, bacteria. And this tissue in dynamic situation, which there is normal ciliary movement to another adverse condition as ciliary stasis. Bordetellapertosis attach strongly to the ciliated cells with the combined action of other adhesins like Fimbriae and Pertoctin. So this bacteria produce toxin, Filamentus hemagglutinin. They have Fimbriae as well as they have a special file was so big, so those slides from last week and this week I put in this PDF and I have sent you. So it is very easy for uploading, that the previous one took time. Pertussis toxin. As we understood, this bacterium can produce a type of toxin with the name of PT, Pertussis toxin. As you see in this photo, 1, 2, 3, 4, 5, 6. There are 1 subunit A monomer and 1, 2, 3, 4, 5 a pentamer B subunit. The most important part is A subunit, and it is enzymatic, and toxicity is related to this part, and the other parts of pentamer in B subunit mostly are working like taxi or shuttle or a car to have this important part to cell. They transfer it from bacterial producer to the target cell. Okay, so they are carrier. These 5 ones are carrier for subunit A and transfer it to the human cytoplasm, which cells, those cells which contain receptors, specific receptors for pentameric B subunit. Pentameric B subunit. And in any cells, if it is matched, the enzymatic or catalytic most important part, we mean subunit A, can be transferred across the cytoplasmic membrane of target human cell to its cytoplasm. And then some things happen. Then some things will be happening, which we aim to understand it. Pertussis, like chlorella and diphtheria, which we explained in previous lectures, is a toxin-mediated disease. Don't have toxin and themselves attack to the tissues, but some other bacteria like Pertussis, they are toxin producers and their toxins attack to the human tissues. However, although Pertussis toxin is important for pathogenesis, it is now clear that pertussis results from the coordinated function of many different virulence factors. So it is saying, it is reminding us, even though toxin is very important virulence factor, even though toxin is very important for pathogenesis, but it is not the only virulence factor, and it is not the only part for pathogenesis. There are several virulence factors are present. The most important one is the Pertussis toxin. As I showed you, Pertussis toxin is an A B5, one A subunit with pentameric B subunit. Virulence factor of the whooping cough. Yes, it is responsible for the disease with the name of whooping cough, causing pathogen Bordetellapertosis, which is produced this toxin by bacteria Pertussis. This toxin can have a behavior as ADP ribosylating. It is a AB5 type toxin and have enzymatic activity as ADP ribosylating or ribosylation. The holo composed of one catalytic subunit. We told you that the central part is the catalytic subunit, is a subunit. 5 membrane binding or transport are B subunits, which form the B pentamer, which are assembled in the periplasmic space and then exported by the forced secretion system. So these are proteins. They are produced by ribosomes in the cytoplasm. The periplasmic space between cytoplasmic membrane, then they are assembled, come together to form a unit, a complete functional unit. Then after this unit is expelled by help of its type 4 secretion system, which is present in the outer membrane of this bacteria. Pertussis toxin, holotoxin, can into almost any sialic acid containing glycoproteins. As I showed you in the previous page, this toxin can go through the cells which cytoplasmic membranes contain receptors. And now I explained that these receptors must contain sialic acid containing glycoproteins. So many different types, as well as lungs, human lungs, contain these types of receptors. And they are disturbed. They are under influence of this toxin according to the presence of these receptors. According to the presence of this receptor. Again, you can see this pentameric B subunits, and in the middle is the A subunit is the catalytic part, is the main part. All the problems are related to this part, and the other parts they find suitable receptor cells. They help to internalize this medium. And when this main part can penetrate to the cytoplasm without need, the helpers do its job, and its job is virulence, is pathogenicity, is stopping the immune system, stopping the Pertussis toxin. You can weight, and we can see in this image that it is hexamer. 5 are related to the B subunit, and 1 is related to the unit. A subunit, enzymatic activity, it can be toxoid, in the factories. It is possible to have some modifications this main part, so the remaining what is remaining from this unit can be toxoid. Toxoid is weak toxin that inducing immune system. So in some cases which there is the possibility for whooping cough by Bordetellapertosis, we can use a vaccine which subunit A is absent or it is malfunctional. It is malfunctional. It lose its enzymatic activity. So immune system can be activated by this toxoid, and accordingly the body be ready reacting to the possible Pertussis toxin is the major component of acellular pertussis vaccine. Okay, page 6. Disability is activate immune system. Yes, it is look like a vaccine. Present only types of bacterium, so toxin can't be found, can't other sources, can bacteria, and is present in this bacterium and is very dangerous. Bordetellapertosis is expressed on the surface surrounding medium. So this toxin is produced in the cell, then transferred to the periplasmic space, and then secreted to the surrounding. If bacteria is inside tubes, flask, or plate, they excrete toxin to. If bacterium is present inside the body of a patient, they produce toxin and excrete inside the patient's body or tissues, causes producing lymphocytosis. This toxin is responsible for lymphocytosis. So the number of lymphocytes are elevated in response of the presence of this bacteria, and can be effective on another metabolic and working cells, like act as histolizing factor and causes excessive insulin secretion. So when in abnormal situation, production of insulin to be higher So when brain, heart, kidney, liver, and most vital parts need sugar, there is not sufficient sugar. And it is hypoglycemia, and it is very dangerous. Maybe or skilled persons. Other toxins like clays. So another toxin is present with work of other cyclase is responsible for overproduction, more production of MP, cyclic in mono enters the target cells and acts as toxin, acts by catalyzing the production of cyclic AMP by various types of cells. And last one, heat labile toxin, cytoplasmic protein present in Bordetella, and thermonecrotic and lethal in mice, was approved by this toxins. Okay, so in this part, after a few minutes talking in general, we want to focus on the mechanism of action of these two types of toxins in our body. You know, our course is microbial toxins and their mechanisms are. So now we want to focus more than or approximately half an hour on mechanism. So Pertussis and 5 membrane binding or transport subunits, the B pentamer. Pertussis toxin is assembled in the bacterial periplasm and exported by type 4 secretion system. Yes, we explained before. It is easy. Toxin has contribution to pathogenicity of Bordetellapertosis by 1. One reason for signs and symptoms and problems in the patient is Pertussis toxin. This is toxin number 1. And some another symptoms are related to the adenylate cyclase toxin. So now we want to focus on 2 different types of toxin, but their actions are. Okay. Now we go through first toxin, Pertussis toxin. What happened? After production of toxin, following binding to sialoglycoprotein host cell receptors, you didn't forget, yes? I told you there are some receptors, and these toxins are produced outside human cells. If they find suitable receptors, following binding to a sialoglycoprotein host cell receptors, then what? Toxin is endocytosed, trafficked through the Golgi apparatus. So endocytosed, going to Golgi apparatus, and by help of this part, going to the endoplasmic reticulum. Here you can see. This is receptor. What is that? Those receptors containing sialoglycoprotein. So penta endosome, and they are covered, they are surrounded, the toxin is surrounded inside endosome. Then endosome transfer toxin to Golgi. Then it is transferred to endoplasmic reticulum. And by help of this part, the catalytic site A subunit is removed from other pentamer, and is released in contact with into the cytoplasm, takes on exosomes to the cytoplasmic membrane. Okay, the A subunit is handled, is transferred to cytoplasmic membrane, where it oscillates the of heterotrimeric protein. So it goes here. Find this heteromeric G protein, a site connected, and this group, by connecting to this, stop immune doing anything by of its stock, stop immune system working. Immune system working. This modification ability of to regulate multiple enzymes and formation. The overall result of this modification is an initial suppression of inflammation and inhibition of recruitment to the site of infection. So it is what is happening by Pertussis Toxin. On another way, by help of another image, we can see again. Don't forget, what is this? This is toxin, A subunit, and pentamer subunit, and this complete unit is Pertussis Toxin. It is Pertussis Toxin. Then what happened? Pertussis Toxin endocyte into a cell, yes, to the target cell. Following intracellular processing, A subunit is created, B subunit. Cytosol. The subunit ADP ribosylates the unit of G proteins, dissociating its output receptor GPCR from this on the cell surface, inhibiting recruitments site of infection. Immune system is of innate immune cells, not allowing bursts to be done by the immune system. It is what we explained in last page and we discussed in this new page. But I told you one another toxin is present by this bacterium with the name of adenylate cyclase toxin. Adenylate cyclase toxin, or ACT. Bordetellapertosis, adenylate cyclase toxin, ACT. So this toxin, unlike the previous one, which was hexamer, this toxin has only two domains. This green one is domain number 1, and whatever is remaining is domain number 2. Domain number 2. So Bordetella adenylate cyclase toxin is composed of two primary domains, 1 and 2. 1. Calmodulin response on host cell membranes from a wide range of cell types. The hydrophobic segments of the linker region form pores in the membrane, enabling the passing of cations, and adenylate cyclase domain is translocated into the cytoplasm. Okay, so calmodulin is coming for helping this phenomenon, and production of cyclic AMP is elevated, and by sending cations out, the action of this toxin can be continued, which is expressed on host cell membranes from a wide range of cell types. The pores in the membrane, the passage of cations, and the adenylate cyclase domain is translocated into the cytoplasm. These two activities are mediated by distinct conformation ACT, adenyl cyclase toxin. Adenylate cyclase activity is stimulated by binding to in the host cell inside in cyclic AMP product. What's at the end in total? The combined of this adenylate cyclase toxin intoxication and pore formation results in complement-dependent phagocytosis, induction of anti-inflammatory cytokines, suppression of pro-inflammatory cytokines, stopping the immune system. It is trying for stopping the job, the work system. Adenylate cyclase with cell surface complement receptor. You can see TR3, that is this part in this figure, on macrophage and are responsible, they have responsibility for attack to the pathogen. So toxins CR3 stop their jobs, affecting antigen presentation and recruitments of downstream adaptive immune response. Adenylate cyclase domain translocates to the cell and is stimulated upon calmodulin binding, leading to increased cyclic AMP levels and accordingly inhibiting pro-cytokine release and complementing mediated phagocytosis, as well as pairing with different types of immune cells. And in the next part, we can see also PT can attack two different types of cell, and here in this section we emphasize that Pertussis toxin can go also from cells growing on ciliated epithelial long cells. Inhibiting migration of lymphocytes across epithelium layers. So lymphocytes wants to go there to do their jobs, production of antibodies and T helpers and the others, but they cannot. They cannot. Why? Because this toxin inhibition. They cannot migrate across epithelial layers. C2 here. Pertussis toxin interferes with GP, which we see in the previous photo, signaling, affecting immune cell recruitment. So stop required for leukocytes, interfering with extra weight. And C4, Pertussis toxin cells. And last page, which we discuss and we finish this part, the inhibition of biofilm formation by toxin, biofilm formation. So in many cases, biofilm formation is beneficial, especially outside the patient, in the environment. Inside the biofilm formation should be decreased. It should be decreased. So when Bordetellapertosis can penetrate to the body, to the patient's body, by help of this toxin, decrease the biofilm formation. Bordetellapertosis has been shown to form biofilm primarily on nasal sputum and trachea. Bordetellapertosis forms biofilm during infection. Recent clinical isolates form more biofilm human infection with Bordetellapertosis hasn't yet been established. It is clear that these organisms produce biofilm both in vitro and in vivo, and thus the regulation of this biofilm formation warrants further investigation. Biofilms are complex structures controlled by they are comprised saccharides, proteins, and extra DNA. Filamentous hemagglutinin, which are produced by these types of bacteria, is an important component of Bordetellapertosis formation. This surface displayed adhesion promotes the formation and maintenance of biofilm by mediating bacteria substrate as well as bacteria by the presence of this toxin decreasing attaching bacteria to the substrate, to the surface, non-living things, as bacteria to themselves to make a big group, big population attached to each other. Although less is known about the regulation of biofilm of Bordetella compared to other medically relevant biofilm forming bacterial species, several modes of regulations have implicated. These toxins active in respiratory because the suitable receptors are there, and according those receptors can attach, can find. Don't forget, all pathogenic bacteria have two main purpose, two main aim. One aim is in the body with their metabolism and according their enzymes can receive food. So number one, they want food, and they do by producing enzymes. They do by producing toxins. Themselves against immune systems. So you see in this toxins are well developed to work to stand in front of immune system and in them done finding food, receiving food, growing, multiplying, as well as stopping attack of the immune system as the enemy for bacteria. If our tissue cells know bacteria as enemy, bacteria know our immune cells as enemy also. So they are the aims of bacteria when they are present in our body. Because the previous group, we reached this part, so, and we want to have parallel progress in two groups. So we finish our short at this point. So next week we continue from 26. We will continue, and there is nothing left. And finish it, and we can next week again. First, I hope health and happiness for all of you along with your family. Thank you very much. See you next week. Goodbye.